Welcome back to our History of Biology and Medicine series. And joining me in co-organizing this series this year is Dr. Sadia Parveen, who is a postdoctoral fellow in CRB2. The series was begun to have experts speak to us about the history of the research topic they work in, what were the earliest ideas that influenced the growth of the field, and where it is now. Today, I'm delighted to introduce Cynthia Sears. She's an expert in foodborne and intestinal infections. She's a professor of medicine and oncology at the Johns Hopkins at our university, and is also the professor of molecular microbiology and immunology at the Bloomberg School of Public Health. Her list of responsibilities is long. Her work has focused on gut infections, including diarrhea and foodborne illnesses during her career. We are really thrilled to have you, Cindy, and welcome you to this series.
Thank you so much, Sarah, and thanks to the Cancer Center for giving me the opportunity to speak to you today. And now I'm going to share my screen. And is everyone seeing my slides, I hope? Yes?
We do.
Oh, great. Okay. So without further ado, let's see. Oops, it's not advancing. Oh, there it goes. And it was stuck, I guess. So first, disclosures. I have grant support to do microbiome work from Bristol Myers Squibb and Janssen, and I am not referencing any off-label drug issues. And today what I hope to do is inform you on the history of the field of microbiome research, or at least give you a flavor for this, and then talk about some of the epidemiology and mechanisms by which the microbiome may impact the pathogenesis and therapy of cancer, and then tell you a little bit about what's in motion about to try to change the microbiome or modify it for clinical care. So this topic is exploded over the last 20 years, making it still a very young field. This has been of interest to scientists, but it's also been of interest to the lay public, and it wouldn't surprise me if some of you get questions about this topic. So if we dial back in time and say, what is the history of this? Well, Robert Hooke in the 1600s, as well as Antony van Leeuwenhoek, were credited with the discovery of microbes. Hooke focused on fungi and had beautiful drawings of the fungal bodies that he published in a book called Micrographia. And intriguingly, Leeuwenhoek, who's credited with discovering bacteria, was actually a textile merchant. And he used lenses to count thread density in his field, and happened to make one trip in his life to London, where he somehow bumped into this monograph by Hooke, and went home and devised an early microscope. And I've always seen him next to something that looked like a microscope to me. So I was surprised to learn that what the early microscope actually was a tiny lens, and then he just put fluid there. So it almost like a microscope slide. And that's where he first saw all those organisms in water. Although he was also credited with discovering red blood cells and sperm and even microscopic nematodes. So he sort of transitioned from being a textile merchant to be a microbiology pioneer. We all know about Pasteur and how he first proposed that diseases were caused by microbes in the 1800s. There was a lot of action in the 1800s, and of course brought us our first food safety approach, pasteurization, as well as sterilization of surgical instruments. And then much has been spoken about Koch, and I'm going to spend a little time on this. He was actually the student of Jacob Henle, and then teamed up with Frederick Loeffler to discuss these ideas about what would constitute causal proof of a microbe causing disease. And as then, he refined these and was the first to publish it. So we know them as Koch's postulates, and we really aren't aware of the other individuals who contributed. Now, Alfred Evans, in a very thoughtful essay written in 1976, talked about the application of these postulates to chronic disease, which of course is what's relevant in cancer, and emphasized a series of points that I think are worthy to keep in mind today as we try to address these questions in the cancer field. And the first is that we really have to understand the correct sequence of exposure in the epidemiology, and then we have to make sure in different populations there's consistency and strength to these associations. We need controls, and the microbiota field has suffered and at different points from this concern to look at specificity. We should consider the immunology and the host response. We need experimental models to test ideas of biologic plausibility, and ultimately we have to see if we can remove the agent or group of agents, I might say, and see if we can prevent the disease. Now, this topic was again revisited just this year in Science in an essay by Brett Findlay and the CFAR group in Canada, talking about whether or not non-communicable diseases are actually communicable. And although this essay didn't focus on cancer, it talked more about cardiovascular disease, IBD, and type 2 diabetes, as well as obesity, which is a leading risk for non-communicable diseases, and pointed out that so far microbial transmission of non-communicable diseases has really only been demonstrated in mice. But here he walked through Koch's postulates, and those include that the pathogen now in the microbiota field, being the dysbiotic microbiota, is found in individuals with communicable disease, and we know that that's true of all these non-communicable diseases I just mentioned. You can isolate the microbe and grow it in culture, and indeed we can do that with the microbiota. It can cause disease when inoculated into a healthy host, and indeed that's what we've seen in the mouse models. And you can re-isolate the group of bacteria from the host, and indeed we can do that. So in a sense, the microbiome field has already achieved Koch's postulates. He goes on to talk about how transmissibility might be happening. And one of the interesting things in the article was he presents some intriguing epidemiology that's emerging, and namely that type 2 diabetes within a year, spouses actually have a higher chance of developing type 2 diabetes that persists for at least a few years. And we know that household members will have more similar microbiota than non-household members. And similarly talks about similar data in inflammatory bowel disease and also the migration effect. Individuals who move from an area with lower rates of IBD and go to higher places, higher areas with IBD develop the disease more commonly. And it's a theme I'm going to come back to. So I would be remiss not to mention to you Colins Toxins, and here's the surgeon himself in this photograph, which was a mixture of organisms that he promoted as a cancer therapy. It's not thought to have any benefit, but I'll remind you of this stunning case of Dr. Brems, which has been presented at the university as, were the bacteria important, or was this a form of immunotherapy? And it was a patient he had in 1991 with a GBM who went through standard care but developed a really intractable infection and remains disease-free 29 years later. Now, that's not necessarily cause and effect, but I heard Dr. Brems present this case in grand rounds years ago, and it has always stuck in my mind. Now, I'm going to show you a paper that's actually one of my favorite papers in the literature. And it's because it combines the concepts of the importance of single bugs and their potential impact on a community, which is a concept integral to the microbiome field. And this paper is from 1970, where Dr. Hastings grew a particular bacteria in a test tube. And after the organism grew to a certain extent, all of a sudden the organism glowed. And in fact, this pivotal experiment is thought to have launched the entire field of quorum sensing, which is the idea that when communities of bacteria are in a group and close enough, they start to impact each other and they release chemicals that actually modify the behavior of the community. Now, the second paper, which is from Margaret McFall-Nagai and Bill Ruby in 1991 in Science, is an amazing story, and they focus on this little animal, the bobtail squid. And what the bobtail squid does, and this is off the coast of Hawaii, filters huge amounts of seawater, which is absolutely loaded with microbes, but only one is capable of colonizing the little animal. So this is an electron micrograph of the light organ of this animal before it's colonized with Vibrio visseri. And here's at 5 days and in 18 days. And of course, you see an evolution. At the end of that period, as shown in the cover of Science, the little animal glows. And it turns out that the bacterium studied here was identical to the organism studied by Dr. Hastings. And so you say, why would the animal want to glow? Well, it turns out it floats in the seawater above its predators. So when the predators look up, they see a little animal that blends in with the night sky. And so it's a protective mechanism of the host. So it's an incredible story of the power of a single organism, but also it led to the incredible observations of how communities differ in their function than single organisms. And I'll come back to that theme along the way. Now, as I said, this is a young field. It's really only existed since the early 2000s. And we always say, you know, the microbiome is critical to health and disease. So I'm going to show you some of the observations and pivotal findings, I think, along the way that really form the concepts we hold fast in the microbiome field. This is a paper from Jeff Gordon, arguably the father of the field, who took a germ-free mouse and then colonized it with conventional flora for 10 days. And this is showing the small intestinal villi and the fluorescein in the capillaries. And you can see colonization with bacteria led to an expansion of these capillaries. And then he did this with a single organism, Bacteroides theta iota micron, and got the same result. So the idea being that microbes are very important to our development. This is a classic paper from David Relman's group, published in Science in 2005, and I'll point out it was only 3 adults, but it elucidated principles that have held up today. And so here they looked at both the mucosal and the fecal microbiota of these individuals using Sanger sequencing, which means that they sequenced nearly 12,000 clones to get to the results, and identified a bunch of novel bacteria in the gut microbiota. At that time, they said they were uncultivatable. This is no longer true. We probably can cultivate at least most of them. The gut microbiota is dominated by the phyla Firmicutes and Bacteroidetes. That remains true. 500 to 1,000. We each carry about 500 to 1,000 bacterial species, and that number has held up as well as principles that were further highlighted in the Human Microbiome Project. Each of us is different, and I'll show that in a moment. And the mucosal and luminal microbiota differ in the colon. Again, an important concept. And this paper from Karen Nelson's lab followed shortly thereafter, introducing the idea of metagenomic sequencing to identify pathways and mechanisms of expression in the gut, or to implicate them, and pointed out that the microbiota provide to us nutrients and capacity that we alone as humans do not possess. I wanted to make a couple comments about the Human Microbiome Project because it's so important to our fundamental knowledge, and two concepts that emerged from this were interpersonal variability and intrapersonal stability. And on the top here is a paper from Paul O'Toole's group in Cork, Ireland, a very important group in the microbiome field, and looking at 161 individuals over the age of 65 in Ireland. And this data shows us how we are each different, whether you're looking at the phylum level, the family level, or at the genus level of the bacterium. So you can just imagine 161 individuals lined up on the x-axis of each of these graphs, and you can see it's a continuum of what each of us has. So we are each unique, but within ourselves, unless we decide to massively change our diet from vegan to meat, we will be relatively stable over time. This paper from Rob Knight's group, another leader in the field and also from the HMP, looked at a relatively small number of subjects, but over time and at many sites. And when they tried to cluster the data by sex or person or day, it was impossible. But what mattered was habitat. And this is this concept of looking at differences in beta diversity, differences between sites in the body. And you can see there's dense clustering of both oral and gut microbiota. But the surprise in this paper, which will be of interest to some, is all these pink dots, which are not so tightly clustered. And these represent areas on the skin. So this paper pointed out that the skin on your foot is very different from the skin on your arm, which was a brand new concept then, and is still one that is pretty fascinating, I think. So I'm going to turn to what we've worked on, colon cancer, and introduce the idea that we actually had long considered that something in the gut, some environmental influence, was important to the development of colon cancer. And this is work dating back to the 1970s. And of course, it's the work where Japanese individuals, Japan having a low rate of colon cancer, when they moved to California or Hawaii, very quickly assumed the rates of disease for a number of conditions, but including colon cancer, that matched the local population. And of course, this led to a lot of thinking about diet and red meat's importance in colon cancer development, for example. And we now know that if you change your diet today, say you decide to become a vegan or go from vegan to red meat, within about 3 days, your microbiome in your gut will be completely different, in classic work done by Gary Wu at University of Pennsylvania. And suggesting that we each have the capacity to have, if you will, a healthier microbiome versus a less healthy microbiome. And these concepts have been reinforced by studies over time with different populations in different countries. So as I mentioned, we work on colon cancer, which is a very important public health problem, not only here in the United States, where we've had some success with screening colonoscopy, but globally. As Westernization across the globe has happened, we've become aware of increasing rates of colon cancer, particularly in Asia. But in data shared with me by Amanda Debes and David Sack in our School of Public Health, even in a setting such as Africa and Tanzania and Zambia, where we think of these as low prevalence areas for colon cancer, there's rising rates of colon cancer. And having looked at this data, this is even involves individuals under the age of 50. And in this country and across the world, we're seeing a bloom of colon cancer in those under the colorectal cancer, in those under 50. And this is now termed early onset colorectal cancer, and fits into the theme of health disparities. And of course, we all know we sadly lost Chadwick Boseman, very talented actor, just a few weeks ago, and just this winter. Trey Mancini, one of the Orioles' young stars, was diagnosed with stage 3 colon cancer at age 29 and just finished his chemotherapy recently. I was fortunate to participate in an NIH workshop on this just a few weeks ago, and the current numbers are that there's an estimated 50 new cases in the United States per day and 10 deaths now per day due to this condition. So I think we can safely say colorectal cancer is a global problem. Now, this is a cartoon I use to continue the theme of the importance of individual organisms versus community. And I'll start on the left. As many of you know, we've long worked with a bacterium called Enterotoxigenic Bacteroides fragilis that, and using animal models, as I'm going to show you, can add this single bacterium to the microbiota of a mouse. And here's the control mouse, and here's with the addition of a single bacterium, and you get this bloom of tumors in the distal colon of the mouse, which I'll come back to. This is not by any means the only organism of interest as single organism in this field, but I'm going to use it as an example today. On the other hand is what we've learned is you take a susceptible mouse who's germ-free and you won't get colon tumors. You'll still get small bowel tumors, but you won't get colon tumors. But if you give back the microbiota, you in fact get tumors in the colon. So this takes me back to an experiment we did now about 20 years ago with Dr. David Houseau, who we sadly lost prematurely in 2016. And our work, which had really been with ETBF as a diarrheal disease pathogen, kept running into cancer pathways. And so Bert Vogelstein actually said I should go talk to David Houseau, and he had min mice that he had created, multiple intestinal neoplasia mice that are APC heterozygous mice. And with David's help, we did this experiment where we colonized the mice with a non-toxigenic Bacteroides fragilis or a toxigenic Bacteroides fragilis and got these remarkable micrographs about 10 weeks later. This was one of the most amazing experiments in my career, and really launched us into considering this whole topic further. Now, Shiguan Wu and Franck Houseau over many years now has taken this forward. And so this is just to tell you sort of where we are today. We know that humans are commonly colonized by Bacteroides fragilis, probably at least 80% of us. And many of us also are carrying these toxigenic strains. What we understand about these bacteria is that the single toxin gene is critical to their pathogenesis. If we take that gene out with an in-frame mutation, there is no tumor biology left. We've learned some about how this works, although there are unanswered questions. We understand that the toxin induces DNA damage in the epithelial cells, that there's activation of STAT3, a pro-carcinogenic transcription factor, and this is a model for IL-17 as pro-carcinogenic. We understand that the IL-17 receptor in the colonocytes is what's critical, but the immune reaction is very important, and it's even asymmetric in the gut, such that there's a myeloid accumulation in the distal gut that accounts for this bloom of tumors distally. Now I'm going to turn to community. and that is, in this case, what we call biofilms. And biofilms are invasive aggregates of bacteria that get into the inner mucus of the colon and then sit right on top of the epithelial cells. And we initially studied sporadic colon cancer. And this cartoon by Angela Chen, a graduate student in the lab, is very helpful to remind us that our human colon is covered with this dense layer of MUC2 dominant, a dense mucus that is largely sterile. It's largely impenetrable. And above it is also a MUC2 layer that's looser and which is invaded by the bacteria. But something happens to allow invasion of that inner mucus. And when that happens, as shown here in micrographs from Julia Drews, you get these beautiful collections of bacteria sitting right on top of the epithelial cells. And on the left here, you can see that empty mucus layer in lighter blue. We've learned that there are at least 3 types of these. So there's polymicrobial, predominantly Bacteroidetes and Firmicutes that make up over half. And then there's an important subset that, as shown in the picture on the right, mostly Bacteroidetes, but then these blooms, these yellow blooms of Fusobacteria. And then there's a small percent that are what we call Proteobacteria dominant, made up mostly of gram-negative rods. We don't know a lot more about how these individually may work or if they're just fully redundant. On the left here is shown a bit about the epidemiology of these, and this is from cohorts in the United States and Malaysia that we studied. And some of this data is through an NCI funded grant we have where Dr. Frank Giardiello is my co-PI. And on the left, you can see tumors. These biofilm positive tumors are highly prevalent in the right colon up to the hepatic flexure, and they're not uncommon in the left colon, particularly in Malaysia. However, from the tumor patient, when you go to the colonoscopy patient who does not have colon cancer, the distribution is throughout the colon and relatively evenly. And we don't know what the critical factors are that push this right-sided dominance. So, of course, that's nice, but does it mean anything? So questions we've asked are, what is the biology behind this? And here we've studied histologically normal colon tissues. And the red stars are just to tell you steps that we've identified in the cascade of how these groups of bacteria might be important to carcinogenesis. So first is these biofilms cause decreased barrier function. And this is just shown here on the right. On the top is a colon cancer patient. On the bottom is a screening colonoscopy patient. Biofilm negative on the right and biofilm positive on the left. And fluorescent staining of ecadherin, a very critical protein to barrier function in the gut. And you can see in the biofilm negative state, there's plenty of ecadherin fluorescence that is quite diminished in the biofilm positive gut. And this is sort of an indicator that there's likely a change in barrier function. There's immune activation, activation of STAT3, which I mentioned, but there's also then proliferation, which is a step before carcinogenesis. That is shown here with Ki67 staining, and on the top is a biofilm negative colonoscopy sample. You can see the brown staining, and the biofilm positive, there's more. You can quantitate all these results, and there's differences. We've also had an opportunity to look at metabolites, initially with Bob Cacero's group, and then with Carolyn Johnson and Gary Zusdak at the Scripps, identifying that there's a specific polyamine metabolite, N1N12 diacetylspermine, that seems to be linked to the biofilms. But that certainly doesn't say that biofilms cause carcinogenesis. So to try to take this a step further, we teamed up with Christian Jobin's lab at the University of Florida, and Sarah Tomkovich, postdoc in his lab, or actually a graduate student at that time in his lab, and Christine De Gea, graduate student in my lab, worked together to ask questions about how these communities might impact carcinogenesis, because at the time we started this work, we didn't have our germ-free facility here at Hopkins. In this set of experiments, we took mucosal tissues that were biofilm positive from cancer patients or colonoscopy patients, and created a mixed slurry that were put into susceptible germ-free MIN mice. And this graph on the right basically shows you that any of the inocula that were biofilm positive, whether from tumors or biopsies or normal flanking tissue of the tumor patients, induced tumors in the mice, but these were not seen in the biofilm negative biopsies used as a control from the colonoscopy population. Now, Julia Drews took on the next challenge, and now junior faculty in infectious diseases, and went back to try to look at individual patients, to try to look at this concept in more detail. And so some of her results are shown here, again, showing colon tumors against a series of samples. And here you can see a negative control. Here is that slurry mix that was studied in the first study. And then here are individual patient mucosal samples put into the mouse model. And you can see this sample 3728T induced a lot of tumors and not so many in the other biofilm positive samples examined. And intriguingly, one of our biofilm negative tumors also induced tumor results. There's a lot to say about that, but that was the basic finding that we were able to replicate in part in specific pathogen-free mice. So Julia went on to culture the 3728T samples, recovering 30 bacteria, and inoculated mice with these, recapitulating the result of the slurry. So we find this to be a very exciting finding because it's now an opportunity to look at a group of bacteria and try to understand what they are contributing to carcinogenesis and how they impact each other. These concepts are not limited to sporadic colon cancer. About 10% of colon cancer is hereditary. This is again work of Christine De Gea with some samples we were fortunate to get from individuals who were having a colectomy due to familial adenomatous polyposis, which is being born as an APC heterozygote. And the two things we really learned here was that this mutation in these patients changed the character of the biofilms, which were present not only on the adenomas, but also in paired normal. And as you can see, there are really only two colors in these pictures. There's red, which reflected E. coli, and green, which reflected Bacteroides fragilis. And as quantified here, the majority of the bacteria we quantified in these biofilms were in fact these two bacteria, very different from the polymicrobial pictures I showed you earlier. The second thing we learned in the red dots are biofilm positive samples was that these biofilms were present throughout the colon, suggesting that once they develop and expand, they really are all over the colon for the most part. Now, Christine was able to drill down, and a bit to my amazement, it turned out that the E. coli was PKS positive E. coli, which has been linked to colon carcinogenesis, and the B. frag proved to be a toxin producing B. frag. And in animal models, we then were able to show that actually there's synergistic interactions between these two bacteria that contribute to colon tumor formation. Now I'm going to turn to big data as a way to do microbiome work. And there's lots of different kinds of big data. As I try to illustrate here, there are clinical databases, and I'm going to show you some data from the UK Clinical Practice Data Link, but there are other major databases that are useful to consider if you choose to do studies. There are a variety of sequencing techniques that can contribute to how we look at the microbiome. And then there are additional methods that are emerging, and I'm going to talk a little bit about computational meta-analysis, but clearly metabolomics and proteomics are going to become increasingly important, as well as likely machine learning and artificial intelligence. And in particular, some view metabolomics as perhaps the common bottom line that we should be focusing on to understand how the microbiome is changing tissue function. So this is a work I want to highlight from Jiajia Zhang here at Hopkins. We had the opportunity to buy data from the Clinical Practice Research Data Link, which is the represents the biggest electronic medical record in the world and is longitudinal data that began in 1989 to the present. So we had the opportunity to look at almost 29,000 colon cancers and 137,000 controls. And what we were focusing on was what was the impact of oral antibiotics on the incidence of colorectal cancer over time. And with the help of the scientists at the CPRD, we came up with a case control study design. The key results are shown here. This is the overall colon results, and what you can see on the x-axis is the cumulative days of antibiotic exposure over the time period that the patients were studied. And the shocking thing to me was that only 15 days of antibiotics, total antibiotics in your life, was sufficient to shift to be associated with a shift in the incidence of colon cancer. When we looked at proximal versus distal colon, all of this change was due to the proximal colon, not the distal colon, which makes sense. If you take oral antibiotics, the carryover into the colon is going to be maximal in the proximal colon. A lot more could be said, but I think that roughly makes sense. We got the opposite result in rectal cancers, where the pace of change was slower, but at about 60 days of total exposure, you actually had a decreased incidence of rectal cancer. Now, one other piece of information from the study that sort of convinced me that maybe this was real, well, there's really two things. One was that when we looked at the timing of antibiotic exposure to the onset of colon cancer, these relationships dated only to antibiotic exposure more than 10 years prior to the cancer diagnosis, a time period that roughly matches how long we think it takes to go from an early clonal expansion of cells in the colon to perhaps a visible polyp. And the second thing was there was a paper published from the Nurses' Health Study almost in parallel that also showed this delayed effect of oral antibiotics on tumor formation. So the two studies were very similar. I'm going to turn now to the other big data approach I mentioned, computational meta-analysis, and this is again work from Julia Drews and James White. And the key feature here is a classic meta-analysis takes the results of the prior paper and analyzes it together with other results from other papers. But here, what we did was we got the raw data, and then we analyzed it through a consistent genomics pipeline, a high resolution pipeline developed by James White. And the difference between this pipeline and others that are commonly used is that it has a much higher accuracy in assigning species assignments compared to CHIME related pipelines. And intriguingly, at the time we did this work, where there were 11 cohorts of colorectal patients, cancer patients published, this is all 16S, rRNA amplicon data, and it's about 500 cancer patients, about almost 400 tumors and 400 controls. And what we found was a set of 5 organisms, many of which from our oral organisms. So we also looked at the human oral microbiome database and found similar correlations. And whether you looked at the difference between healthy samples and cancer samples, or looked at normal tissue versus colon cancer patients, you got exactly the same set of organisms. And when we look back at our cohort, we in fact have similar results. However, I will say I don't think we're going to sequence our way to any answers that are durable for patients in the microbiome world. And I use these two papers, which are excellent papers in Nature Medicine about a year ago, looked at a huge number of fecal metagenomes from colon cancer patients, identifying a small number of species associated with colon cancer and a sensitivity of about 80%. But the issue was they either didn't look at adenomas or they failed with adenomas. So while this was highlighted as a way for translation, the truth is if you had a positive test with this, you needed a colonoscopy. And if you had a negative test, you needed a colonoscopy. You can't miss 20% of cancers. So I think we really are going to have to complement sequencing approaches with biology and other approaches. Nonetheless, this is a very useful data set, and we're actually currently trying to get all these metagenomes. Now, I've talked about colon cancer, but this is a topic relevant across the cancer world, and I'm going to point out two papers. This year, one was in Science in May from the Reisman, where they looked at 7 different tumors and looked at the bacterial populations. And as you know, at least 16% of overall cancer incidence is now attributed to microbes. And here are just some of the key results. One of the things that was excellent about this paper is they had over 500 normal samples, over 1,000 tumors, and they had 811 contamination controls, often missing in microbiome. So they really went to the mat to try to deal with contamination because these are likely low biomass types of analyses, which is where contamination can be really important. And here is just shown the bacteria through 16S rRNA PCR, the levels detected. Here are the controls on the left. Here is melanoma, and then I'm going to point out GBM. And at the highest level was breast. Now I'm going to show you one more graph in this paper, and this is the number of species detected per sample. And on average, it was less than 9, except for breast, where it was an average of 16 species. So personally, I struggle a little with the low rates in melanoma, the presence in GBM, and I don't have an issue with the breast data, but it strikes me as unusual and you know, how to interpret that. They did do a lot of other experiments. They did cultures, which tended to correlate with their 16S rRNA data. They did imaging, which I did not think was all that impressive. And they did PRI-CRUST-2 analyses, which is a way of using sequence data to look at function that was more impressive. Now, I'm not sure, you know, I have questions in my mind about these data, but Rob Knight's group, nearly in parallel, published very, very similar results using an entirely different approach, looking at the TCGA. So I think this bears paying attention to and should give us all things to think about in terms of looking at patients within our center. Now, as just a couple more things I want to share with you. These are 3 signature papers you're probably tired of hearing about, and that is the potential contribution of the microbiome to the efficacy of immune checkpoint inhibitor therapy. The problem with these papers and a few others is they all tended to get different results, and they all tended to use different analytical methods. So I want to highlight the importance of team science in microbiome work, and because of the BKI, we have been fortunate enough to assemble a team from clinical faculty to clinic staff to laboratory staff to try to look at some of these microbiome questions, and that also includes our germ-free facility. We are still in the early phases of this, but I wanted to highlight the work of Faisa Shaikik, who just joined our faculty, working with James White, who did a computational meta-analysis of the studies I just showed you, as well as others that had been published. And now she's again looking at the raw data, and we got a very different answer. And that answer was, in red are bacteria associated with non-response. In blue are bacteria associated with response to checkpoint inhibitor therapy. And so this was the flip of how these results were originally published in the literature. And I just, in red is highlighted that the non-response bacteria gave a sensitivity about 70%, whereas you can see the response bacteria were not so impressive. Faisa went on to look at some validation cohorts, finding she could validate results in melanoma cohort that had been subsequently published, as well as, to our surprise, hepatic carcinoma, but not in lung cancer, which I think is a tougher one. So this is a piece of what we're trying to develop further. So I just want to mention a few other things. There are many challenges in microbiome disease analyses. I've mentioned the contamination issue, and this is a paper by Curtis Huntenhauer, working with 15 laboratories, a set of samples, and indeed they all got many different results amongst these different labs. So it's something we have to be aware of. This recent paper in Science, I think, brings to our attention that we have probably not thought broadly enough about the types of exposures that may be impacting the microbiome and disease genesis. And this excellent discussion from Fergus Shanahan, again from Cork, sort of taking issue with all the positive rodent experiments and the fact that we don't have translatable products in the clinic, is a good read. And he argues for more rigor and critical consideration of these data. So what can we do? Well, there are many, many clinical trials underway addressing a broad range of ideas, from bacteriophages to sort of recolonization of patients. But these are all very much in motion, and I think we have to wait to see what happens. What's a little bit closer and happens sort of real time for us is fecal microbiota therapy, which because of COVID is largely on hold and was first described in 1958. This is a problem. We have seen serial FDA alerts with transmission of multi-drug resistant organisms, specific pathogens, and even deaths now, including a description in the New England Journal. And I think we have to open our minds to consider some additional approaches for patients. I'll just point out this recent study from Sears that uses spore-based therapy, which are predominantly from firmicutes, and showed a very impressive decrease in CVI recurrence. We're probably going to see this emerge on the market in the foreseeable future. I know I get asked a lot about probiotics. I wouldn't be surprised if you don't also. And I just want to point out, again, this literature is weak at baseline, but there have been some really good papers recently that have pointed out, and this is from Aaron Elanoff's group in Israel, that probiotics can actually slow down resolution and re-establishment of a microbiota in the gut. And their lactobacillus is the #1 probiotic used across the globe, and yet the New England Journal published recently 2 completely failed trials with probiotics in children that had a probiotic that had been endorsed from the WHO down. And I'll lastly point out this paper, which I found intriguing in Nature Biology just last month. Again, looking at this issue of trying to fix our microbiota after antibiotic exposure, identifying tiers of organisms that seem to build on each other to allow the microbiota to recover. And I think it's a concept, again, we should give some thought to. And of course, I'm deeply indebted to our funders, as well as everyone who has worked in the lab and all of our internal and external collaborators. And with that, I'll be happy to take questions. And here is your CME code if you need it. So thank you very much.
Thank you, Cindy. That was a really interesting and enlightening talk. I will start off the questions. What was intriguing to me was the story you told about populations that have immigrated to this country, yes? And that same story is told about breast cancer as well, that the population of Japanese who came into the California area actually had incidence of breast cancer equaling that of USA, although the incidence in their home country was so low. So I was wondering, and the first change that takes place is lifestyle, particularly food. So did you find common bacteria that seem to predispose individuals to breast and colon cancer?
So that's a great question. And I actually didn't try to look at their data to try to answer that question. I assume those data sets are deposited. I would think that different tissues will have different bacteria. Although of note, some of the breast literature has included Bacteroides, and this is something, as you know, Dipali Sharma and her group is really working on, and which we've been helping with a little bit. I do think there could be connections, but I'm not sure there are connections. I think that needs a more careful look. It's a great question.
The other thing is that, of course, the gut and the microbiome are like living with each other, and therefore the interactions are very intimate compared to the distant organs. The other question, and much more particular, was the question of you had biofilm positive and biofilm negative, and there was one patient in one category and one patient in the other category who had a number of bacteria. Now, what did they have, the non-biofilm patients, that the biofilm patients did not or did?
Yeah, it's a great question. Again, that is very recent data, and we're just in the process of trying to answer that question, but I don't have an answer yet.
All right, let me open the forum to others to ask questions. And please unmute yourself, or you can ask it in the chat box. I will present, this is Linda Resar's question. Is homozygous loss of IL-17 required, or is there a phenotype with heterozygous loss of IL-17 signaling? There she is.
Yeah, hi. Yeah, hey, Linda. Our experiments were done with knockout of IL-17. Okay. And I don't think we actually did the same experiments with heterozygous IL-17. But if Frank Houseau is on the call, he can correct me. I don't recall that we did that.
Yeah, and I'm sure you can guess why I asked that question.
Yeah, I know exactly why you asked that question. Yes. And, it's an important idea that, just a little muting of the signal may still have gene interactions, just like SNPs, may have gene interactions with microbiota. Yeah, thank you.
And there's a question from Jenna Canzoniero. Are you on board? You want to ask it yourself?
Well, I can read it. It says,
oh, yes, go ahead, please.
I can see it. Can you please comment more on the effect of the role of antibiotics in the food chain? I imagine that the usage of antibiotics in farming may also be different between countries, and I wonder if that contributes along with diet differences. So again, an excellent question. I don't know of precise data from the food chain, but there certainly is hints that introduction of particular additives may then have impact. Now, the farming thing is a fascinating story because, as you know, antibiotics are added to feed in about the 1950s because it made chickens plumper. And so they brought more money at market. And now Marty Blazer's group has shown that just low levels of antibiotics actually will make a mouse get plumper. So suggesting that, in fact, this type of change and transmission may actually affect biology. And that is part of the thrust of a lot of work now in the obesity field, is to look at how this may be impacting children and how that may impact disease onset, not only obesity, but things like atopy and asthma over time. So again, a great question. And the other place this is, of course, very relevant is antimicrobial resistance. And Europe was ahead of us in pulling out antibiotics and actually seeing antimicrobial resistance rates go down. And we're finding, and we're reversing some of that here now. It's not perfect, but it's in progress.
Any other questions from the attendees? If not, may I ask you one more, Cindy? And
Sure.
that is to do with ear infections. Now, as kids grow up, they undergo multiple cycles of this exposure. Have you found out whether children in this country whether this exposure has some impact on developing cancer versus other countries that don't use it this often?
Yeah, so that, again, that's a great question. This is one of the targets of the early onset colon cancer field, where in fact, there are cases of colon cancer, sporadic colon cancer, not hereditary, in teenagers that people are seeing are very young adults. So there is concern that, you know, the generation affected mostly by this fell into that group where we know we were overusing antibiotics. Diets changed. It was part of the obesity epidemic time. And it feels like those are relevant risk factors. We still know that, you know, about 30% of our antibiotic use in the United States is probably not necessary. And so there are a lot of campaigns on to try to deal with that. Obviously, when antibiotics are needed, they should be given, but we have to become probably more discriminatory in the outpatient setting in particular.
Any other questions? Otherwise, I'll pose one more that's been on my mind, and that is that probably the easiest or the simplest way to change the microbiome would be diet. So people have said that the Mediterranean diet is almost the best for preventing cancer, which is where we should be headed to. So is there any move in that direction? Because we are, as a people, quite receptive to ideas that help us to live healthier lives, most of us.
You know, I think the answer is yes. I mean, that is a widely publicized concept. Helping people to do that is a little bit of a challenge in a country that is really experiencing food threats, and in urban areas where we have food deserts. So we shouldn't have such tremendous food challenges in this country, but I believe we do. There are a couple in the chat if you want me to try to address.
Please do.
From Joe Murray, in patients with COPD, is there data to support differences in exacerbation, antibiotic usage, and risk of progression to lung cancer? I do not know of those data if they exist. Again, a great question. And then there is a question, if I could comment more on the lactobacillus probiotic, was not successful in FMT, considering that lactobacillus is a dominant species in the female reproductive tract. So what I was talking, the lactobacillus trials that were done were for 2 strains. that is associated with, used widely across the globe as a probiotic, and had been thought through a lot of observational studies to decrease acute diarrhea in children. So that was the context for examination in two large studies, one done in the United States, one done in Canada. And I think it was a surprise to all of us that there was absolutely no single signal of benefit in those studies, which I think should all make us pause before we recommend current over-the-counter probiotics. Whether we can design something that will be effective remains to be seen.
Also, in intestinal bowel disease, have you seen E. coli PKS to be present?
So actually, E. coli PKS was discovered in the context of ileal Crohn's disease. That's where that organism first really emerged as linked to disease. They are also very common, particularly in children, and they have no disease association there. So this is another concept in the early onset colon cancer field, is there are the enterotoxigenic B. fragiles are probably most common in young children. The E. coli PKS are very common in young children. We don't know from whence they came and when they emerged like that. And so do these organisms and their communities play a role in early onset colon cancer?
Thank you. I think there's one more question here.
Diarrhea and gases would be simpler symptom to follow. Not quite sure what the question is. I think in the trials I mentioned, they were trying to shorten the duration of diarrhea, since dehydration is a major killer in young children in developing countries.
So with that, we will thank you very much, Cindy, for really a beautiful talk and a beautiful history of an emerging field. Thanks, everybody, for attending.
Thank you very much, and thank for everyone for their attention. Have a great day.
Have a great day.
Bye-bye.
Bye.